All right, so what we are going to do though is talk about a little more detail about phototransduction. So the actual conversion of light energy into the electrical signals. And again, which are the cells that do the phototransduction?
The rods.
It's going to be rods and cones. The basic process is the same, whether it's a rod or a cone. There are some differences in the exact, energies that are being absorbed for the different colors. There's different, we'll talk about different kinds of opsins. We saw there's difference in just the wiring in the retina, whether it's one ganglion cell per cone or many rods converging on a single ganglion cell. But the basic idea of converting light into electrical signal is the same, whether you're looking at a rod or a cone. That's what we're going to look at right now. So let's look at the classic rod. They're kind of weird looking cells. They have what's called the outer segment. This is connecting down. This is like to the bipolar cells. So going down to the rest of the retina. So if you remember, when we drew the retina, it was rods and cones feeding into bipolar cells, feeding down into the ganglion cells. So these are just kind of puffing out like they release glutamate. Yeah, I should mention the neurons in the retina, except for the ganglion cells, they don't actually have action potentials. What they have are these graded potentials that increase or decrease the amount of neurotransmitter being released. But it's still these signals. So let's start in the dark. Let's assume there's no light. If we're in the dark, there is a sodium channel that's actually open. Which means that the cell is going to be more positive than resting. And we call it like the dark current. And the membrane voltage is around minus 40 millivolts. So it's actually going to be a little bit backwards from what your intuition might be. In the dark, the cell is actually above threshold and it's releasing glutamate to the next layers of the retina. But it doesn't matter. All we care is that there's going to be a change. So it turns out the change is going to be when the light comes in, we're going to go lower the voltage. The voltage is going to go down below the threshold. So basically, the rest of the retina knows if glutamate is being released, that means we're in the dark. If the glutamate stops, it means light happened. So this dark current is happening from, obviously, there's got to be some channel. This sodium channel is held open by cyclic GMP. So it's going to be a second messenger pathway that ends up making cyclic GMP. So as long as there's cyclic GMP in the cell, the sodium channels are open. And the sodium channels are open, the voltage goes up, and we've got this dark current, and we got this membrane voltage around -40. Does anybody remember what we could add to the mix to get rid of the cyclic GMP?
Is it phosphodiesterase? No.
Yeah, no, that's exactly it. So ultimately what's going to happen is light is going to end up turning on phosphodiesterase, PDE, which is going to close these channels and is going to lower that voltage. And that's going to be the signal. That's going to be the transduction. What we're going to talk about now is how is it that light ends up turning on phosphodiesterase to turn off the dark current? And that is going to be some other membrane proteins up in the outer segment. So up in the outer segment, we got to pay attention to a membrane protein called opsin. Opsin, which is connected to another molecule called retinal. They form a complex. And then there is a G protein called transducin. Most of the time we just call G proteins, but this was like one of the first of these second messenger pathways people came up with. And it was like, oh, it's helping transduce, and they called it transducin. So it gets its own name, but you can also just think of it as a G protein. So what we need to do here is talk about opsin and retinal, because they're the things that actually act as kind of the receptor for light. And the retinal is kind of like the antenna that absorbs the light. Or actually, I should say the opsin and the retinal together are like the antenna that absorb the light. And when they absorb the light, it's going to activate the G protein, which is going to ultimately turn on a second messenger pathway to make phosphodiesterase. So let me focus a little more on opsin and retinal. So here's my let me make sure I use the same color so I don't confuse you. I had opsin green, retinal red. And here was our G protein. So in the dark, this complex just kind of sits and it's just doing it. So it's just sitting there quiet. If light comes in, though, retinal has a conformational change. This part is just you have to accept that this retinal opsin complex is kind of like this antenna that can absorb light, and it absorbs it at the frequency that that particular photoreceptor responds to. So depending on the type of opsin, it might be absorbing green light or blue light or red light to make this happen. But when the proper light comes in and is absorbed, that energy actually is used to change the shape of the retinal. So normally when we're in the dark, we have what's called cisretinal. And when light is absorbed, it flips into what's called transretinal. And that is going to be the beginning of this kind of chain reaction that's going to result in our phototransduction. So we're in the dark. Everything was like I mentioned here. We got the dark current. We have minus 40 millivolts, which is actually kind of positive, right? That's above threshold. We have this complex here, kind of waiting, nothing's happened yet. Now light has came in. Maybe I'll write it out. I'm gonna give myself some space here so I can actually write this. I'm gonna think how to do this in a way that I can be efficient. So like in the dark, that's probably too small. In the dark, there's that sodium current, due to channels held open by cyclic GMP, and VM is around minus 40 millivolts. So that's in the dark. That's where we're kind of starting. Now light comes. So the retinal converts from cis to trans. So that retinal, which is up here now, is going to flip. And when that happens, it actually detaches from the opsin. And that's what basically activates that system to turn on the G protein. So this is, instead of normally you think of a receptor binding some ligand and turning on a G protein. In this case, when the retinal absorbs the light and has this conformational change, that activates this opsin, which is basically like this receptor, and it turns on the G protein. And what happens when you turn on a G protein?
Second messenger pathway.
Yeah, so it activates the second messenger pathway. And I'll just say that ultimately turns on PDE, right? Phosphodiesterase. And then what's going to happen once we have phosphodiesterase turned on in the cell?
The cell will close. The channel, I'm sorry, the channel will close.
Yep, so sodium channels close. And VM goes down to more like minus 70 millivolts. This is the actual phototransduction. Maybe I should change the color here. So when the sodium's channel closed, because the phosphodiesterase is for the phosphodiesterase chews up the cyclic GMP, which has been holding the channels open. The channels now close and the voltage goes down. That's the change in voltage. That is the phototransduction. The voltage was minus 40 and it changed to minus 70. So we had light and it created a voltage change. Does that make sense? The voltage change kind of goes in the opposite direction that you might think of intuitively, but it's still a voltage change that the rest of the retina understands as light having hit the retina.
I have a question.
Sure.
You talked about PDE earlier in the class as kind of like the secret sauce in Viagra.
Well, no, Viagra is it inhibits PDE.
Okay, but it affects it, and it's a major player in that area. Is it kind of found having these various very different roles throughout the body? Or are these two of the
No,
main?
there are many different subtypes of PDE even. And there are all these second messenger pathways that use cyclic AMP or cyclic GMP usually involve it in some way or another.
Okay.
So yeah, there are so many different things in the body mediated by second messenger pathways, and erection is one, and phototransduction is another. And then there's a bunch of lots of other things as well. And drugs that, they try to, like Viagra works on a particular subtype that is just, or that is part of the erection pathway, but it does have side effects. I think it actually does kind of affect your vision to some degree in terms of shifting things bluey or something. It's most drugs that you take almost always, they do what you want them to do, but then they're not magic bullets. You take aspirin, right? You take aspirin for a headache. It's inhibiting prostaglandins. It's also, you take aspirin for a fever and it's taking down your fever. It doesn't know you want to take it for a fever or for a headache, or it doesn't know you're trying to take it for inflammation because you've got tendinitis. It doesn't know why you're taking it. It's doing all those things all at once. And all you're kind of paying attention to mainly is what is the relief from the symptom that you were worried about. But everything else that it does is also happening. It's not like well behaved to just, so usually that would be my answer, is that you're focusing on one primary, you're taking it for a particular indication and you're focusing on, is it doing that? But it's probably doing lots of other things as well that you're just not noticing. So, all right. So I do know that this is kind of convoluted, but it should feel relatively familiar because you've already got to know convoluted second messenger pathways and stuff like that. So are there any questions about this process? So
I have one more question.
sure.
On this drawing, which on which side of it is the retinal wall?
The back of the retina is, let me get, this is like, hold on. This is the back of the eyeball.
Okay. So the bipolar cells in that direction, that just, that faces out into the middle of the eyeball and toward the front.
Yeah, this is looking towards the front, towards the pupil.
Okay. Thank you.
Sure. Again, one of the kind of weird things about the eye, again, when light's coming in here, here's the lens, the retina is on the back. All of these cells that we're talking about are facing towards the back of the eyeball, which is non-intuitive, totally non-intuitive. The rods and the cones that I was just drawing, they're like here, facing towards the back. And then you have, bipolar cells are the next cells. Then the ganglion cells are the next cells. And then their axons are making the optic nerve. But luckily, so the light has to come in, it has to go through all of this, all these cell bodies to finally get to where the phototransduction happens. But luckily, cells are transparent. So they're, and also at the fovea centralis, this is more of an anatomy thing, but you basically have the cell bodies are kind of pushed off to the side to kind of thin out the thickness of the retina. So you have minimal distortion at the fovea centralis. At the fovea centralis, it actually looks like a little divot because all the cell bodies are kind of squished to the side to minimize the thickness and the distortion. You look at cephalopods like squids and octopi, they have cooler eyes than us in a lot of ways because the rods and cones are facing the front. If you look at a squid or an octopus, the rods and cones are facing towards the front, which means two things. One is there's no level of distortion through all the cell bodies. And what else is the benefit of this?
Don't have a blind spot, right?
There's no blind spot. You don't have to have some place where they have to go burrow out the back from the front. So there's no blind spot and there's no distortion. But again, we're not going to get so into it, but the way ours are set up is the rods and cones have their heads buried in this called the pigmented layer where they can be maintained, and the photopigments are getting replenished and all sorts of stuff. So, you know, the way our eyeball is set up, the rods and cones get to be like, have nice, you know, intimate access to their metabolic support on the back of the eyeball there.
Are both the rods and cones, they both are attached to bipolar cells?
Uh-huh.
Oh,
Yeah. And again, this idea of bleaching, like in really bright light, most of the retinal in the rods has turned into cysts. So when you first turn off the light, most of your rods aren't working yet. You have to wait until that, sometimes they call it rhodopsin or visual purple even, because when they first started tearing apart animal retinas, they'd see this purple stuff that then would bleach in light. It was actually like, you know, the opsin. But so it takes a while when you turn down the light for the rods to kind of replenish their retinal opsin complex and start working properly. So that's that kind of why you have to wait a few minutes when you get into the dim light to really see well. And also why, if you turn on the bright light, you destroy your night vision for a while, because then it's going to take another 5 minutes to kind of get that back going again. All right, so last thing I want to talk about vision is kind of how the two eyes are hooked up into the brain. So let's think about, here's a person, this is supposed to be their nose and their eyeballs. And they've got a brain, here's their brain in their head, two hemispheres. And we talked about how the right hemisphere deals with the left half and the left hemisphere deals with the right half. For vision, the hemispheres are dealing with half of your visual field. So if I am looking at this half of my vision, this half of my visual field, kind of drawing in green. This is kind of landing on this side of both my eyeballs. So I want to have information from both eyeballs from that right side of the visual field here. And that's going to go to the left hemisphere of my brain. So I get messages from both eyeballs about this left side of my visual field. And there's no, this is the right side of my visual field. This is the left. So the left side of my visual field over here is coming in onto this other side of my eyeball here. So both eyes are sending information about that side of the visual field to the opposite hemisphere. And that's where you get that optic chiasma. right? It's a really obvious X of the optic nerve when you look at how the optic nerves enter the brain, because each eye is sending half of its fibers to one hemisphere and half of to the other hemisphere. Right, and what is the benefit of having two different eyes versions of the same visual field?
Depth of field, perspective.
Yeah, that depth perception that we saw. Has anybody done the thing yet with like putting a pencil into a little thing? Nobody?
I tried, but I feel like I didn't, like it was the same. I don't know if I wasn't using a jar that was small enough.
I mean, you want to do something that just fits the pencil. So something more like a test tube kind of shape. So yeah, I would do it again, but with some, I'm trying to, yeah, you need to do it with something where if you're off by an inch or two, like if you try using a big jar and you're off by an inch, you're probably still going to get in the jar. So that's not quite.
Yeah, I use like the smallest jar I have, but I don't think it was small enough.
Oh, so
Like a water bottle work.
well, you know what you might do is something like a pen. You know, a pen cap. Can you put like a pen cap would be a great thing. Like if I have my binocular vision and I try to put a pencil in a pen cap, I can do it. But if I have one eye, like I literally tried there and I messed up, right? So something like a pen cap that from a Sharpie would be a great way to do it. So something more like that. So, you know, not all animals have their eyes like that. In fact, like what's the downside of having both eyes on the front of your face?
Can't see behind you.
Yeah, it's pretty easy to sneak up behind you, right? Which would be bad if you're like a little more of a prey animal, you know, and you really are worried. Is there like something coming sneaking up behind you that wants you for lunch? You are more likely to have your eyes on either side of your head, right? And then your field of vision, instead of being kind of like 100 and something degrees in front of you, is going to be more like 360 degrees all around you. You know, it's really hard to sneak up behind this guy. He's like, doo-doo-doo. He's like, you know, whoa, right? He can notice that this kind of predator is coming up, even though he's like not looking at him, because his field of view actually goes pretty much 360 degrees all the way around him. This guy, it's easier to sneak up behind him probably, but you know, most predators have their eyes in the front of their head because they really are using that depth perception to capture their prey. Like, you know, this dude's hanging out. It's like, doo-doo-doo. His brain is calculating exactly where is this squirrel. And in one fell swoop, it's like, got him, right? He knows exactly where he has to lunge to get his dinner. So in general, predators tend to have their eyes on the front of their head for that. You know, think about a cat versus a mouse, right? You know, things, or a hawk versus a sparrow or something that eats meat versus eats seeds. You know, in general, most animals, yeah. So, you know, we're omnivores. We definitely are predators. So it's nice having that depth perception, so we know exactly where it is to catch our dinner. Anyway, so that, I think that's where I'm going to leave vision. Okay.